So I'm really pleased to be here this afternoon to help introduce Dr. Karen Tanner. Dr. Karen is a banana slug like me in that we both did our doctoral work at the University of California, Santa Cruz. So yay, go slugs. And when she worked there, she partnered with Elkhorn Slough National Estuarine Research Reserve to test strategies aimed at improving salt marsh restoration outcomes. That, of course, is really important to SFBBO since we do work on salt marsh and transitional habitat restoration. During this time, she also investigated the impact of solar energy development on desert annual plants for the California Energy Commission. After earning her degree, Karen began a California State Sea Grant Fellowship at the San Francisco Bay Conservation and Development Commission, where she supported the agency's work on sea level rise planning for the last year. In her spare time, she loves to hike and pee out native plants. So hopefully when I'm back in the Bay Area, I can get Karen to go on a hike with me because I love hiking with people who know plants because I'm really bad with plants. So without any more from me, I just want to hand it over to Karen and thank her for coming and doing this talk for us. And also thank you again for joining us.
Thank you, Yue. It's such a delight to be here and speak to all of you today. We are going to talk about salt marsh restoration and whether positive interactions between plants can have a positive outcome on restoration. And I want to just acknowledge first the collaborators that I've had on this project. There's actually probably even more than the folks that are on this list, but this is one of the projects in particular that I'll speak to you about is quite large with a lot of different people working on it. So without further ado, okay, so many of the dunes and wetlands that were once present along California's coast have been replaced by agricultural fields or by urban development. And in fact, we've probably lost more than 90% of our North American coastal salt marsh just in the last century. It's really often not possible to turn that clock back because there's just limited opportunities in these kinds of settings to do restoration work. But coastal wetlands are really high priority targets for restoration efforts where they can be done because they buffer sea level rise and they also sequester carbon that would otherwise fuel climate change. On the West Coast, as many of you may know, the largest remaining salt marshes that are still around are found in San Francisco Bay and in Monterey Bay. But even here, the losses have been extensive. The shoreline around San Francisco Bay is highly developed, providing relatively few opportunities for large-scale salt marsh restoration. The South Bay salt ponds, shown on the top right, are an exception, and I believe that SFBBO has been a really active participant in some of the restoration efforts in this area. In Monterey Bay, the Elkhorn Slough Tidal Wetland Program and its partners have been working to improve degraded marsh and restore land that was diked and drained for agricultural use. If we could travel back in time 130 years to look at the Elkhorn estuary, the main channel would have been surrounded by extensive salt marsh habitat. We've got it shown here on this historic map in green. And the mudflats, which are shown in brown, would have really been fairly limited. So these are areas that just experience too much tidal flooding to support any salt marsh plants. If you put them there, they would just drown. By the year 2000, the estuary had changed dramatically, with over 1,000 acres of historic salt marsh lost to land use change and habitat erosion. To combat this ongoing marsh loss, the Elkhorn Slough Tidal Wetland Program was formed. Its purpose is to develop and implement strategies to conserve and restore estuarine habitats in the watershed. And they have a variety of goals, which I've got a short list, which is incomplete on the screen right now. And I think we will, Serena, if you can cue up the video, we're going to watch a short video on the Hester Marsh restoration, which is one of the most recent Tidal Wetland Program projects.
The wetlands of the Elkhorn Slough are home to animals large and small. They filter water flowing down from the hillsides and hold carbon, an important benefit in combating climate change. But the slough has lost 50% of its salt marsh in recent decades. With the Hester Marsh Restoration Project, we're working to get some of that back.
And the Hester Marsh restoration is really an attempt to sort of set things back into balance. We are not here to recreate the past. There's been changes made here that we'll never be able to turn back, and these changes have affected this place pretty dramatically. So what we are really trying to do is create something new that is sustainable well into the future.
The Hester Marsh Project restores 65 acres of former salt marsh that was lost due to historical diking and draining. The first step in restoring the sunken land was to bring in soil to fill it back up.
The way this project was designed, we moved about 200,000 cubic yards of sediment out here. We had to raise the salt marsh plain about 3 feet in order to make it at an elevation that's high enough to sustain sea level rise, the modeled sea level rise that we anticipate seeing.
Heavy equipment rolled and rumbled constantly for over 8 months, hauling in soil to raise the elevation, then grading and sculpting the land.
The contractors understood the conditions out here, how to work in the wet mud, and how to create what you see now.
Once the stage was finally set, the new channels were open to the tidewaters. This milestone marked the end of construction and the beginning of life returning to this once and future marsh.
What we're seeing today is the public coming out and having a chance to get their hands dirty in this project and contribute to this restoration effort, because it's not just one group or a few people that are doing this. It takes an entire community to make this happen.
By studying historical marshes and healthy wetlands, and by monitoring changes here, like the rate of plant growth, the return of wildlife, and the amount of carbon held in the soil, we are learning a lot about how these habitats respond to disturbances like human activities and climate change.
When we have a project on this scale, there are huge opportunities to learn from it. And we will be studying this project for many years with diverse teams of scientists and volunteers. And the lessons learned will inform not just our own work here at Elkhorn Slough for future projects, but also salt marsh work around the world.
We're restoring 65 acres of salt marsh ecosystem. We're improving water quality through filtration. We're sequestering carbon. We're providing resilience to future sea level rise. And we're providing habitat for our listed species such as otters.
Within a few years, we expect the marsh to be lush with vegetation, providing a refuge for wildlife, and enough soil was added for the new marsh to sustain a century of sea level rise.
Great, thank you, Serena. So as we saw in that video, there's really a lot of work and a lot of decision making that has to happen as part of these marsh restoration efforts. And to ensure that these decisions are made with the best available science, there's more than 60 scientists from a variety of disciplines that make up the Tidal Wetland Program Science Panel. And the TWP is also charged with fundraising so that it can implement on-the-ground restoration projects like Hester Marsh, and also the restoration of Whistle Stop Lagoon, which is another project that I'm going to talk about today. And just to give you a sense of where I fit into this picture, I am one of the grant-funded researchers that's contributing to the program's decision-making process by carrying out field experiments and reporting on the results. I partnered with the scientists at the Elkhorn Slough National Estuarine Research Reserve to develop and test planting strategies that could improve early restoration outcomes, when we're really most concerned with getting that new native salt marsh vegetation established. Salt marshes are considered stressful environments, especially in the summer when we really receive little to no rainfall and the salts can become really concentrated in the soil from repeated cycles of tidal inundation and evaporation. Clustered plantings have been advocated by some as a tool to reduce stress and increase plant survival and growth. I'll walk you through why clustered plantings might be helpful, and I'll tell you what we've learned from testing this approach. We usually think more about plants competing with each other for light, for nutrients, or for space, kind of like what we imagine is happening in this picture of the tropics. But there are situations where neighbors can be beneficial. For example, this cactus got its start under a nurse tree where the desert temperatures were less extreme. There is a classic ecological framework called the stress gradient hypothesis that predicts neighboring plants can provide benefits when growing conditions are stressful. The hypothesis predicts that when physical stress is low, plants will tend to compete and they'll suppress each other, just like what we imagine happening in the crowded tropics, where temperatures are mild and water is plentiful. But when conditions are stressful, like in the desert, neighboring plants can buffer that stress. For this saguaro cactus, lower temperatures under that tree canopy allowed it to survive through the seedling stage and make it to adulthood. This idea has important implications for restoration in stressful systems. In the low marsh, where we get a lot of tidal inundation, there is some evidence that clustered plantings relieve waterlogging and also erosion stress. Let's just take a moment to imagine a grouping of plants versus a single isolated plant. And then imagine that, both of those are exposed to the same swift current. You can kind of imagine that the grouping of plants will probably fare better. They're just going to be able to resist that force better together. And that's the kind of benefit that neighbors can provide in the low marsh. But what about the high marsh? The physical stressors in the high marsh are much different. Tidal inundation is much less frequent here, and plants are really only underwater about maybe 1 to 7% of the time, depending on their elevation. So waterlogging and erosion aren't really major stressors in this zone. However, during our dry Mediterranean summers, evaporation concentrates the salts that are deposited by infrequent tides. And you can see a sort of whitish haze on the soil surface. So could clustering be used to relieve drought and salinity stress in high marsh restoration projects? We tested clustered plantings as a restoration strategy at 2 high marsh restoration sites, Whistle Stop Lagoon and Hester Marsh. We'll start with Whistle Stop Lagoon, which was diked off, drained, and used for agriculture for many years, as a lot of our coastal wetlands have been. The Tidal Wetland Program funded the installation of a box culvert into this lagoon in 2014, and that immediately restored the natural tides into the system and improved the water quality drastically. And that seawater inundation also killed back the salt intolerant upland plants that had sort of crept down into the tidal zone over time. And when that happened, it left behind a bathtub ring of bare soil that's visible in the photograph on the left. Now, native salt marsh plants were really slow to colonize this bathtub ring, and we didn't really know why. The only thing that was coming in was the native pickleweed, which is often the dominant vegetation in marshes in Monterey Bay that have undergone tidal restriction, like this lagoon did. And other natives like Frankinia salina and Jamea carnosa, those can be up to 25% of native cover elsewhere in this watershed, but they remain virtually absent at Whistle Stop, and we really didn't know why. But one thing that we did notice is that when you do see these natives in the Elkhorn estuary, they often occur in these large patches, kind of within a surrounding sea of pickleweed. So that was an interesting natural history observation because it suggests that maybe there's something beneficial about growing in clusters for these two species. And we wondered, could positive interactions between plants be responsible for the patterns that we see in the field? Is there something about having neighbors that helps these native plants? Can we imagine a scenario where neighbors may be beneficial in the high marsh? Let's think for a minute about our Mediterranean climate, where desiccation can be a really big risk for seedlings. In bare soil, which is what we had in that bathtub ring at Whistle Stop, temperatures are higher and moisture loss is much more rapid. It might be helpful to have neighbors if a grouping of plants shades the soil better, keeping temperatures and evaporation rates much lower. If conditions are less stressful next to existing plants, then it might be easier for additional plants to establish nearby. And if that's the case, then outward spread from sort of a central established location could lead to the development of large patches just like those we see in the Elkhorn estuary. So these natural history observations suggested that clustering might work for us as a restoration tool in the high marsh. At Whistle Stop, we know there's an elevation gradient from the low marsh to the upland, where the tides and evaporation create gradients in both moisture and salinity. Our restoration effort was focused on the high marsh, where the moisture stress really starts to increase. And you can see it with your eye when you're out there in the field. The ground starts to look really kind of hazy and it gets all cracked. Looks like a rough place to try and grow as a plant. So we might expect neighbors to be beneficial as we move upslope on this elevation gradient. Clustering plants together might improve survival and growth of transplants that are closest to that upland zone where the conditions are going to be driest. We use loose and tight planting treatments to test whether clustering can mitigate this stress. In the loose pattern, our neighboring plants are separated by 50 centimeters, and then plants are only separated by 10 centimeters in the tight planting. So the idea is that, in the tight planting, the plants are close enough together that they can interact and that they can shade each other. But the plants in the loose planting are too far apart really to affect one another. We predicted that loosely spaced plants should do better at lower elevation where moisture stress is low, and that clustered plants should do better at high elevation where it's drier. We planted both treatments using Frankenia or Jamea at 91 plots across this elevation gradient at the lagoon. And we tracked plant survival, drought stress, plant growth, and also cover. But how do we know that the physical growing conditions at higher elevation actually were more stressful? We thought that was the case, but we really needed to do some kind of an investigation to show that this was true. So what we did is we measured soil water potential across elevation as a proxy for drought stress. And I want to walk you through what water potential is and why it matters to plants. Water potential is measured in megapascals, and 0 is actually the maximum value possible, what you'd see if you measured distilled water. In contrast, seawater from the estuary has a lot of dissolved salts and other materials in it, and it has a water potential of negative 2.89 megapascals. So as we move upslope, the soil becomes drier and also saltier due to high evaporation rates in summertime. So as we move upslope, we might expect to see a pattern like this for bare soil. We see that the water potential is getting lower and lower as the soil gets drier and drier while we move up the hill. But for soil that's under a plant canopy, where those soil temperatures and evaporation rates are lower, there might not be as much change across elevation. And that's what we wanted to find out. So this is how better shading of the soil by groupings of plants could potentially provide a benefit. So what we did for the actual measurements, we took a soil core at a bare location in each plot, and that is where we expected the soil moisture loss to be greatest. And then we took a second core underneath the plant canopy, where temperatures and evaporation rates should be much lower. We used a water potential meter to measure these soil cores and compare what they had to say about the conditions in the field. So this instrument can be used to measure plant tissue as well. So it's actually a really nice way to get measurements on both the soil and on the plants that are planted in that soil in a common currency. So you can see if the soil and the plant tissue are showing you the same patterns across elevation. I'm going to orient you to this plot before I show you the data for the soil. So if you look on the bottom of the screen, you can see that elevation is shown on the on the x-axis. And then if you look on the left side of the screen, you'll see that water potential in megapascals is shown on the y-axis. And then I've got as a reference point distilled water, which remember has no salts in it and has the maximum possible water potential value of 0. That's indicated by that blue dashed line. And then as the soil becomes drier or saltier, your water potential is going to become more and more negative, which means it should be more and more difficult for plants to extract any moisture that is present in the soil. Okay, so here's the data, the first set of data, which is for soil in the open. Each triangle shows the water potential of a core taken in the open. And you can see there's a clear decline in water potential as we move upslope. So these data do suggest that plants at higher elevations should have more difficulty extracting any moisture that's present from the soil. But for soil cores taken underneath those plant canopies, shown here by the filled triangles, the decline is much less pronounced as you move upslope. So this is good news. This is what we expected. We found a strong gradient in soil water potential across elevation during the dry season. And we found that canopy shade slowed that decline, suggesting that clustering could reduce stress at bare earth restoration sites. Okay, but did our plants respond to this measured change in soil water potential across elevation? We have a strong gradient, but do the plants care? I'm going to walk you through 4 different ways to sort of look at and think about plant performance: tissue water potential, survival, plant growth, and percent cover. And in each case, we'll consider whether plant performance changes across elevation as we might expect it to do based on the soil measurements that we took, and whether clustering improves performance as you move upslope. We're going to start with tissue water potential and transplant survival. So the way that we did the tissue water potential is we took samples from the central plant at each plot within 24 hours of those soil cores that we took, just cutting stems right off that central plant and taking it back to the lab. On the top, I'm showing you a mock-up of the pattern in tissue water potential that we should see if plants track the trends we observed for soil water potential measurements. So in this mock-up data, the open circles show imaginary plants from loose plots, and the filled circles show plants from tight plots. So first we'll look at the data for plants from the loose plots for each species. So these patterns that you're seeing on the bottom of the screen, shown by the pink and yellow circles, they do not look like the pattern at the top of the slide, right? They don't match our mock-up. So we don't see tissue water potential declining across elevation like we saw for soil water potential. That's interesting. Nor do we see the expected pattern in tight plots, shown by the filled pink and yellow circles here. And if we just totally ignore elevation for a minute and instead just look for a difference in tissue water potential between the loose and the tight planting patterns, we don't see a significant difference for either species. Tissue water potential is about the same, whether the plant tissue sample came from a loose plot or a tight plot, it doesn't matter. Hardly any of our transplants died either. So we had about 94% survival at 8 months after planting. And survival did not differ between the loose and the tight plots for either species. You can see that those bars are just about the same height in either case. Okay, so let's review what we know so far. So remember that for each species, we want to know if plant performance changed across elevation. And then we also wanted to know whether clustering had an effect on tissue water potential or survival. So this summary slide provides us with some columns where we can keep score. We can track elevation and we can track clustering effects for each species. So for Frankinia, there was no effect of elevation on tissue water potential or on survival. And there was also no effect of clustering. And we saw the same thing for Jamea. So far we have no evidence that plant stress increases with elevation as we expected it to, or that clustering helps reduce that stress. But what about effects on plant growth and plant cover? 6 months into the experiment, counted the number of stems on each focal plant. And let me tell you, that was a lot of work because the plants grew a ton in that period. So here the top photos are showing you what the plants look like just right after we planted them. It was probably the same day or certainly within the same week. And the bottom photos were probably only taken about 2 to 2.5 months later. It was a real struggle to count those stems, which in a way is great because you want to have your transplants just thrive. It just made the data collection a little difficult is all. So here we're going to look at the average number of stems per plant from that survey, and that's what's shown on the y-axis at the left. And again, we've got elevation on the x-axis at the bottom, so we can see if that's changing across elevation. And I'm going to have open circles showing loose plots and filled circles showing tight plots. Frankinia stem counts increased with elevation in the loose plots, shown by this dashed pink line and the filled, or I'm sorry, the open circles. But we didn't see that pattern in the tight plots. It looked like there really was not much of an effect, or maybe a slight decline in stem counts as you moved upslope. In contrast, elevation had no effect on Jamea, regardless of the planting pattern. You can see as elevation increases from 1.7 to 2.2 on the x-axis, there's really no systematic increase in the number of stems that we counted. And again, if we ignore elevation for a minute and just look at things in terms of the loose pattern versus the tight pattern, stem counts were significantly lower in the tight plots for both species. So this suggests that close neighbors are actually competitively suppressing the focal individuals at the center of the planting. That's not what we were expecting to see. So let's add these results to our summary table. For Frankinia, elevation effects depended on the planting pattern, but there was no elevation effect for Jamea. And clustering had a negative effect on stem counts for both of these species. Okay, but what about effects on plant cover? This is really the coin of the realm in restoration projects. It's usually how we set goals and also how we measure outcomes and the overall success of the project. What we want to do is track progress from bare ground at a newly restored site to fully developed mature marsh vegetation. Does clustering help us achieve better plant cover? Or does it help us get the same amount of cover faster? Okay, after 8 months of growth, we collected percent cover data in the plots to determine how much of that bare ground was covered by Frankinia or Jalmea using each planting pattern. Percent cover is shown on the y-axis here, and elevation, as usual, is on the x-axis at the bottom for each species. Loose plots, again, are going to be open circles. Tight plots will be the filled circles. Frankinia cover increased with elevation only in the loose plots, shown by the dashed pink line and the open circles. And elevation had no effect on Jalmea cover, regardless of planting pattern. But if you look at those clusters of yellow circles, you can probably see that on average, the tight plots, which are shown by the filled circles, they have slightly lower cover values than the loose plots. I can't resist showing you these photographs, even though we just looked at figures that encapsulate all of the data across all of the plots, because photographs are just so great for delivering the message effectively. So all 5 of our transplants are visible in the loose plots on the left, but you can no longer distinguish those individuals in the tight plots on the right. And you can also see with your eye that there is less transplant biomass overall in those tight plots. So we'll add percent cover results to our summary of effects. Frankinia cover increased with elevation in the loose plots only, while elevation had no effect on Jalmea cover. Clustering had a negative effect on cover for both species, again indicating competition rather than the facilitation we were looking for. So these patterns are the exact opposite of what we'd expect based on our soil water potential measurements. Why is this happening? We need to consider rainfall conditions. So this experiment was planted in February 2017. So what I did was look at the historical rainfall record from January to June and compared that to rainfall during our experimental period. So what this figure is showing is every blue bar is the rainfall from a single year, and there are 61 years here, that's the historical record, arranged in order of increasing rainfall. So each of those blue bars is a year, but it's not chronological, it's arranged in order of increasing rainfall. And then the 5th, the 50th, and the 95th percentile cutoffs are just provided to give you a sense of what low, average, and high rainfall years look like in this system. And here's 2017, right? So rainfall was very high in 2017, and our transplant survival data also showed that it was an unusually good year. Remember, we only had about 6% transplant mortality, and that is excellent for a restoration project. I'm not sure I've ever been involved with another restoration project that had that good of a survival rate. But we know from earlier work that's been done in the Elkhorn system that transplant mortality for these very same species can be quite high. Anyway, we let the experiment run on for another year and returned to assess percent cover one more time in fall 2019. So we've got our percent cover on the y-axis, and as usual, elevation on the x-axis. Let's see what Frankinia did. So by fall 2019, I saw clear patterns in cover across elevation. But the interesting thing was that they differed between these two species. So Frankinia cover increased with elevation, while Jalmea cover declined. This is mysterious. and very interesting. These inverse patterns are very interesting to us because it suggests that there is something that's changing across elevation in a way that does affect these two species, and it is affecting them differently. But we still saw no evidence of facilitation between plants at the less favorable end of the elevation gradient. Regardless of the planting treatment, Frankinia still did better at higher elevation, and Jalmea did better at lower elevation. So where does that leave us? Our first goal out there was to identify the location of greatest stress in the high marsh. And we did measure a really strong gradient in soil water potential that suggested plant stress should increase with elevation. We then tested whether clustered plantings reduce stress at that location. But we found little evidence that plant performance tracked changes in soil water potential across elevation. We found that clustering always suppressed plant growth and cover. So these results suggest that clustered plantings are not a good tool for improving restoration outcomes in the high marsh, at least with these two species in a high rainfall year. So what would give us more confidence in these results? What would give us a better sense that these patterns are real and repeatable and that we can rely on them? It would be useful to know how plant interactions unfold in different weather years. 2017 was excellent rainfall year. What about something more around average or something that's, a low rainfall year? There is the potential that there would be a different outcome. And it would also be good to know if other marsh natives show the same patterns. And it would be really great to know if these patterns also manifest when restoration is carried out at a much larger scale. So let's shift gears now and talk a little bit about Hester Marsh, which was the subject of the video we watched. As you saw in the video, sediment was added to this restoration site to raise the subsided marsh plain about 2 to 3 feet. And this is what the site looked like in fall 2018. It was just this vast expanse of totally bare dirt. We weren't ready to give up on clustering quite yet, although we talked about it, because we had had such a high rainfall year for the whistle-stop restoration, and because we had 5 native species that we could test at Hester Marsh. We thought clustering might be beneficial at these larger restoration scales because these really vast expanses of bare soil experience even higher soil temperatures and evaporation rates than the small scale plots that I was working in at Whistle-Stop. So if clustering could help any of these 5 native species under these circumstances, it could still be a useful restoration tool for us. On this map, the color shading indicates elevation, and that pink zone on the left, that represents the high marsh. So the same zone that we've been working in all along. We had large blocks in the high marsh at this site where we planted 5 native species in either a uniform or a clustered layout. Let's look at just one of these blocks to see the uniform and clustered layout in more detail. So within a block, there's a pair of columns for each of the 5 species, with one of those columns planted in a clustered layout and the other column planted in a uniform layout. So the total number of plants is the same in both of these columns. It's just a question of how they're arranged. I mentioned that we wanted to know whether clustering helps in a drier year. But as fate would have it, we planted this site in another high rainfall year. So what have we been seeing out there so far? This photo shows our starting point, which was February 2019, when we all got super muddy and also got rained on quite a bit. And I love this picture. It's so clear. It so clearly shows what's going on out there. So this is definitely one of those pictures that's worth 1,000 words. So a uniform column is shown on the left and a clustered column is shown on the right. On the top, we're looking at April 2019. On the bottom, we're looking at August 2019. And it's already pretty clear, right, that transplant biomass was higher in the uniform column. And you can also see that there's just a lot more bare soil in the clustered column on the right. So what I'm showing you right here is Frankinia because it photographs well, but we also saw this pattern with Extraplex and Spargularia. The Jomea and the Disticlus have been slower growers for us at this site, so they didn't have really clear differences in 2019 by planting pattern. But in 2020, we did the same survey and we were starting to see some differences with more biomass in the uniform planting pattern for those two species as well. I wanted to also mention that all this work that I've just described was part of Hester Marsh phase 1. So phase 2 of this restoration project is underway right now. And we've taken what we've learned from these experiments. We've decided like, well, it maybe is still possible that under dry circumstances, you could see a benefit of clustering. We've tried it twice and we haven't found it twice. So we are not currently planning to do additional testing of this strategy at this location. But there are a great number of research projects that are still running out there, looking at everything from water quality to carbon sequestration rates, as well as habitat use by otters, birds, insects, invertebrates. And I really cannot stress strongly enough, if you have not visited Elkhorn Slough and or Moss Landing, it is such a great place to visit. And I highly recommend, if you're able to rent a kayak in Moss Landing and then just paddle up the estuary, that you'll see so many otters, so many birds, so much just marsh habitat is really quite wonderful. So with that, thank you so much for your attention. And I just wanted to acknowledge all these additional people beyond the collaborators that were on the first slide, who were like critical to getting this work done. And I would be happy to take any questions that you may have.
Awesome. Thank you so much, Karen. That was fascinating. I love to see things like, experimental plots, things happening in the field. And yeah, the results were surprising. And I do see a question. Could it be that Frankenia is more adapted to higher saline or higher stress than Jamea? And how would this compare to other non-restored sites?
Yeah, that's a great question. So one of the things that I tried to look at with the whistle stop work was, different salt tolerant plants can have and do have different adaptive mechanisms for handling excess salt. And so Frankenia and Jamea, there doesn't seem to be a lot of knowledge, detailed knowledge about exactly how Jamea is handling its excess salt, but it doesn't excrete it. Frankenia, on the other hand, has these glands in the leaves, and those glands, if you've ever been out in a salt marsh, like in the morning when it's kind of cloudy and you like sort of touch the Frankenia, you'll get like a salty film on your hands. And that's because it has these specialized like salt excretion glands in the leaves. And so that's how it gets rid of excess salt. It doesn't try to exclude it at the roots. It lets it come on in and it just like pumps it right on out again from through the leaves. So there's definitely, I cannot answer the question directly of, is it more adapted, but it definitely has a different salt tolerant physiological mechanism through which it's coping with that situation. And it definitely does seem like I would be curious if like in the San Francisco Bay and the work that SFBBO has done, if like you see the same sort of general patterns, but down in the Monterey Bay, we definitely do tend to see Frankenia salina at the upper end of the high marsh. That's where it looks happiest. It can exist lower, but it's most commonly found at the higher end, whereas Jamea is a little bit lower. So it does seem that they sort of are self-sorting into like their preferred, their preferred zone. So in terms of like, how does it compare to sites around the rest of the estuary? One of the challenges that we face, and I'm sure it's probably the same in San Francisco Bay, is we just don't really have like a lot of like, we have sites that haven't been heavily disturbed in like maybe 100 years, but we have like almost no sites that have never been disturbed. So it's always so difficult to like figure out like, well, what is your reference? Like what are you gonna look, if you wanna know like what a truly intact undisturbed salt marsh looks like, what do you look at, right? And down at Elkhorn, we, I don't think there might be like one or a couple spots I can think of where it probably hasn't been disturbed in 100 years, but the rest of it is all kind of undergone a variety of impacts that have come and gone. Sometimes it was diked off and then, the levee was breached and, just there's just been a variety of conditions that have led to a variety of different assemblages of vegetation. And that's one of the things that we would hope eventually to try and tease out through the work that the Tidal Wetland Program project, all those projects that it's funding and looking at is like, you know, can we figure out like, you know, I started by saying Whistle Stop Lagoon underwent tidal restriction, and then we didn't have Frankinia salina and Jamaica, so we just had pickleweed. You know, that seems to be a general pattern. When we have tidal restriction, we have lower diversity of marsh plants. So that's one of the reasons that we really wanted to go to the effort of outplanting those less common plants at Hester. I hope that answered the question. That was a little bit long-winded.
That was great. But related to diversity, there is another question. Is there any historical record of how these plants grew in the past? And do they require to be in a community of specific multiple plant species?
I don't know. I am not personally familiar with the details of like how, I know that there has been research done on this in Elkhorn Slough, like historically, you know, what was the composition of the vegetation like? But I can't unfortunately speak directly to that because I just wasn't part of that work, so I don't know it. But it is true that you know, for the salt marsh, like the high marsh zone that I'm talking about, pretty much what you see there are like the 5 natives that I showed photographs of in the Hester Marsh section of the talk. So it's Frankinia salina, Jamea carnosa, Disticus spicata, Spergularia macrotheca, and I feel like I'm forgetting somebody. Who is it? Anyway, Extraplex californica. And there are also non-native species that we have. So, you know, in that high marsh zone, for things to really persist, like for any length of time, they have to kind of be halophytes. They have to be salt tolerant to some degree or other. So you don't, you know, the high marsh isn't as heavily invaded as like the adjacent upland. And I'm sure it's probably the same in San Francisco Bay because it's just sort of a restricted suite of plants that can really cope with those conditions. We do have a few halophyte exotics. We've got, you know, rabbitfoot grass, we've got Achiplex prostrata, we've got probably at least another 4 or 5, you know, you can get ice plant, you know, there's stuff that comes in. But one nice thing about the high marsh is that, like, especially compared to California, like grassland uplands that are above the high marsh, they're like way less invaded comparatively. So it's a nice, kind of a nice place to spend your time, except that of course, you know, 5 feet away, you've got a wall of Brassica nigra or poison hemlock or whatever. So.
Karen, I want to ask you, any last things you want to share with everyone before we sign off?
Just to reiterate, to like go down to Moss Landing, and if you can get one of those kayaks, like if you go up the main channel, you actually will end up passing the Hester Marsh restoration site. It's like not very far up on the right-hand side, so you could potentially go and see it. You could see, even right now, there's all this heavy equipment moving the sediment around, so it would be easy to spot. Go outside and have fun.
Nice. Yeah, we did have several people mention that they hadn't been back to that area in many years, so I'm sure it'll be interesting to see how it's changed. So yeah, thank you so much, Karen, for your wonderful talk, and thank you everyone for joining us.